My name is Stefan Herlitzer and I'm the PI of Project A07.
My name is Katharina Spoider and I'm the Associate Investigator. In our Project A07 we aim to understand how stress and serotonin interact to modulate extinction learning. Using an optogenetic approach in combination with the cell-type specific activation of individual serotonin receptors, we try to get insight into the modulatory actions of serotonin during normal and stressed conditions. Stefan, there are several key structures in the brain involved in the formation and processing of fear memory, like the hippocampus, the medial prefrontal cortex, and of course the amygdala. And as far as I know, most of them are innervated by serotonergic fibers. So what is actually the main focus of Project A07?
Kata. We will concentrate on the influence of the serotonergic system on information processing in the amygdala, which plays an important role in storing emotional memory, such as fear memory and fear memory extinction. The amygdala consists of various nuclei and cell types. The relevant simplified main circuit consists of a basolateral and a central part. The basolateral amygdala receives both direct and highly processed information from all modalities. This information is processed locally by inhibitory interneurons and then transmitted in an excitatory manner to the central nucleus.
The output of the central nucleus is inhibitory and projects to hypothalamic and brainstem areas that mediate the autonomic and behavioral signs of emotion. Fear conditioning has been extensively studied in the amygdala. Most studies use electric food shocks as an unconditioned stimulus paired with a conditioned auditory stimulus. Auditory and nociceptive information converge in the basolateral amygdala and induce plasticity changes within the network, most likely underlying memory formation.
Fear responses are in particular increased by stress, and stress increases the level of monoaminergic modulatory transmitters in the brain such as noradrenaline, dopamine, and serotonin. Serotonin in the brain mediates its effect via serotonin receptors. Two of them are very highly expressed in the basolateral amygdala, that is the 5-HT2A and the 5-HT2C receptor. Thus, in this grant, we would like to understand the role of the serotonergic transmitter system for fear and extinction learning in stressed and normal mice.
In the first set of experiments, we will control the release of serotonin in the dorsal nucleus and in the amygdala during fear conditioning, using an optogenetic approach, and the part will help us to identify the critical phases during which serotonin can modulate the learning process and how this depends on stress.
Since serotonin acts on 5-HT2 receptors, and alteration in the receptor expression have been described during stress and fear conditioning, we would then analyze if the distribution and function of these receptors changes during the learning process in the basolateral amygdala using immunohistochemistry and electrophysiological studies.
In the last aim, we will analyze if the optogenetic control of 5-HT2A and 2C receptors is sufficient to modulate the alterations induced by serotonin during fear conditioning. We will therefore make use of the chimeric light-activated GPCRs, which we developed to control 5-HT2 signals by light. We have used this approach before to control anxiety behavior in mice.
Thus, our work will lead to the understanding of how serotonin modulates fear and extinction memory during stress and normal conditions.
In addition, we will learn how two important drug targets, the 5-HT2A and 2C receptors, modulate and maybe directly influence fear learning and extinction.
So we are both very excited about our Project A07, which we describe in detail in our grant application.
And finally, we hope we could raise your interest and enthusiasm for Project A07. Bye-bye.
Bye-bye.